Button here. All right. And so as Bridget mentioned, if you guys have questions during the webinar today, by all means, please send send it via the chat box. So I'll write something into the chat box right now and send it to everyone. And what should happen is on your end, it should light up kind of with an orange kind of color highlighting your little Zoom banner. And you will see that there's a chat option, and you should see my message written there that just says hello everyone. And that will be the box that you can use during today's webinar. Okay, and I'll periodically stop and ask, if you guys have any questions or anything like that. But in the meantime, let's go ahead and get started. So I always get a lot of questions on, why should we use these tools? I'm so used to doing my, experiments, just creating a series of manual gates and coming up with the different populations, measuring their MFIs, measuring their frequencies, and then making, sort of head-to-head comparisons between tubes or between groups of tubes against each other. There's nothing wrong with, analysis that way. And of course, ultimately, that's what we end up doing in most of our experiments anyway. The problem comes in when we try to analyze highly complex data in a short amount of time. And so as we add more reagents, right, let's say we start with a 4-color panel and we jump from 4 colors to 12 colors, we have only a 3-fold increase in the number of, in the number of parameters that we're looking at, or sorry, 4-fold. But what ends up happening is the number of 2x2 plots that you now have to go through begins to grow exponentially. And so you can imagine that, from 4 to 12 colors, okay, if 12 colors is, you're getting bogged down a little bit in terms of the analysis. When you go from 12 colors to 20 colors and 20 to 40, it gets even more complicated. And these tools, both dimension reduction and clustering, have been designed to help you reduce the analysis burden. They are there to help you visualize the data and to make comparisons between, your healthy and disease, or your, wild type versus knockout, et cetera. And so really all of this has been brought on by the advent of new cytometers, as well as new reagents, and of course, new software tools. So some of you may have heard of the Aurora from Cytec. That's you can basically run 40 color panels on those machines. BD has a Symphony A5, I believe, that's also right around the 40 color panel range. And then Sony also has a multicolor or multi-panel, multicolor spectral cytometer. And so all of these things have sort of contributed to adding more markers, as many markers as we possibly can onto the surface of the cell and try to look at it via flow cytometry. We also do this in sequencing, in that we use antibodies that have been tagged with oligos, and you can dump a heck of a lot more antibodies onto cells in that instance, because you don't have to deal with fluorescent spillover, et cetera. But even in those experiments, you have to use these tools because the complexity of the experiment is so great that you have to use something to simplify the information. And so generally speaking, all of this stuff, all of these tools kind of help to alleviate the burden that we have here of going through really complicated data. All the possible combinations can be kind of mind-blowing. But ultimately, what we want to do is, when we're analyzing these complex data, that the idea is that we're going to, it's possible that we are going to find a novel population of cells, or perhaps it's a known population of cells that we're looking at, but in this particular disease instance, they're doing something that we are unaware of that particular function. And so ultimately you want to be able to get back to the cell sorting step. And the question then is, well, how can I go from, this complex data, digest it down, and then get to the final step of sorting? So the high dimensional workflow is kind of laid out for you here, and I'll go kind of over each of the little bubbles here, just in a little bit of detail here and there, not too much, because we'll jump into FlowJo here proper. But I think it's very important to understand what the order of operations is when you are starting to look at complex data. And I see there's a couple of things in the chat box here. So I see. I am in present. Oh, you would like presenter view for the PowerPoints. I will do that. Yeah, I apologize. I tend to just use the PowerPoint and skip from slide to slide, but this is much better, so you guys can see it a little bit clearer. Okay, so I think, as I mentioned, it's pretty important to use these tools for highly complex data, but it's important to understand how to do the analysis and what are the appropriate steps. So the first bubble here that's sort of in the top right is the data quality control, data QC step. This is the same for any flow cytometry experiment that you're going to come across, right? You have to inspect the compensation matrix that's on your cells. You have to get rid of doublets, debris, things of that nature, dead cells, because you don't want to feed dirty data essentially into these algorithms, because they're going to, look at those dead cells and debris, and they're going to discover, a bunch of novel populations of cells that you've never seen before, and it's going to cause you to go on a wild goose chase. So you want to do data QC first, clean up the data, inspect the compensation matrix. The compensation is particularly important because, if you have one sample out of 10 that has a compensation problem, and then you feed those 10 samples to these algorithms, that one weird sample is going to look significantly different from the other samples. And again, you're going to have kind of a hard time figuring out what has gone wrong or why I see all these novel populations of cells when in reality they're just poorly compensated cells from that one tube. So again, it's very important to make sure that your compensation is good before moving on to these tools. Okay, so after data QC, there are some tools available for normalization of the data. Normalization is a great idea. I don't know that the normalization question has really been answered well in flow cytometry, but there are tools out there to help you reduce batch effects, for example. And if you have you have a longitudinal study and you want to make comparisons between, time point 1 month, 6 months, 1 year, and let's say 2 years down the line, you want to be able to normalize the data such that the biological effects are still present, but the batch effects or the day-to-day, variance of the machine is eliminated. There is a tool called cytonorm that we have available in FlowJo to help you normalize the data. Some people also that may have different cytometers in different locations. So somebody in, San Diego, another person in New York, they may have the exact same machine running the exact same panel, and they want to make comparisons, let's say, between those two patients. Normalization would be very important for that type of a setup. And again, cytonorm is what FlowJo has. There's a couple of other R-based tools that are out there as well. They're freely available, so definitely worth giving it a shot. After the normalization step, I have annotation. I have to say that annotating the data once you are, when you're getting ready to use all of these high dimensional tools, taking the time to annotate the data is going to save you a lot of time. And when I say a lot of time, I literally mean like hours of time on your analysis. So if you assume that you've got a 25 color panel and it's going to take you 2 hours to 2 hours to finish the analysis just by manual gating and so on and so forth, the comparison stage is going to take you another probably 4 to 6 hours to complete. But if you annotate the data, that's going to reduce that time down to probably 1 hour or less. So there's a huge benefit to annotating the data at the beginning, so that making comparisons between groups is very easy later on. And I'll show you how to do that as well, and we'll actually make use of the annotations to compare the data. The concatenation step is the step where we merge all of the files that we wish to compare to one another in this when we use these high D tools like dimension reduction and clustering. We want to concatenate the files first so that they're projected onto the same space, and that way it allows us to make comparisons between samples. So the concatenation step is generally the primary bifurcation point between your standard analysis and the analysis that we're going to do with high dimension. Okay, so concatenation is what makes this webinar very different, I would say, from other webinars, plus some of the tools. Obviously, the tools are very different, but it is an incredibly important step for being able to make comparisons between samples. If you don't do concatenation, dimension reduction, clustering, and all this downstream stuff is going to be either very difficult or almost meaningless. So concatenation is what we need to do, and I'll show you how to do that as well. Okay, dimension reduction, there's several different tools that we have available for dimension reduction. These are tools that do exactly what the title says. They reduce the numbers of dimensions required to visualize the data. So you've probably seen a t-SNE plot like I have here, or this actually is a UMAP, the big bubble here. And each blob sort of corresponds to a different subset or different phenotype of cells, and it projects it all into a single two-dimensional space for you. So you can see how your cells are changing based on a given treatment, right? Let's say you treat the drug, cells with a drug, on day 1, and then you want to see what happens to them on day 5, whatever. You will be able to visualize that easily using dimension reduction tools. Okay, clustering tools are usually used in conjunction with dimension reduction. They perform sort of a similar function in that they divvy up the data into different phenotypes of cells, but rather than giving you a visual output, they just spit out populations for you in the workspace. And clustering algorithms are good for essentially 2 things, in my opinion. Number one, they were developed so that they could analyze the data or crunch the data very quickly. So generally speaking, clustering algorithms are going to identify populations of cells in your data set within minutes versus hours of setting up a series of manual gates. So for speed, that was the number one reason they were developed. But number 2, clustering algorithms have a unique ability to identify cells that I would say the human eye is not necessarily privy to. And what I mean by that is a lot of mid-expressing cells, right? Any marker that is sort of mid-expressed, or cells that are sort of moving along some sort of trajectory, clustering algorithms tend to pick those out a lot better than the human eye does when it comes to manual gating. So I like clustering algorithms for sort of giving you this fresh perspective on the data, find those mid-expressors. Ultimately, we'll have to figure out, whether or not those mid-expressors are significantly different than the ones that are high or low, but the point is the algorithm sees them and pulls them out for you. Okay, so after you do the dimension reduction and clustering, normally what we're going to want to do is some phenotyping of those clusters, whether they be in the t-SNE space, or if we use a clustering algorithm like Phenograph or FlowSum, for example, we're probably going to want to figure out what those populations are. Once we ID the phenotypes, we are going to want to make comparisons. This is obviously where the money is at, right? We want to be able to see which cells are changing as a result of our drug treatment or the disease condition, et cetera. And so we're going to do a comparison step after we do some basic phenotyping. I also have a step here for tracking. You probably, if you have a longitudinal study, you're going to want to see how those populations change over time. Maybe not just in the context of disease versus not disease, but maybe you have a situation where a patient goes in pre-operation or pre-treatment. You see what their cells look like, and then you want to take a look. Let's say a week after their operation, and then maybe I don't know, 3 months after the operation, and a year after the operation, et cetera. And being able to track the cells to see how they move over time is going to be an interesting path. So I'll kind of introduce the topic here when we get to it, and then ultimately we want to be able to isolate those new populations of cells that we find, or even the ones that we are known to us, but we want to be able to bring that back to the sorter. So I'll show you how we can do that. Now I have to escape out of here to because there's a couple of slides that I want to skip over because they're not really pertinent to what we're going to do here, and that has to do with all of this stuff. I really want to just jump to the t-SNE algorithms and so forth, and just give you a quick primer on these before we kind of jump into FlowJo proper. So I'll put this back in presenter mode. Okay, so there's several different dimension reduction techniques that we can use, and I always get this question, why should I use t-SNE versus UMAP versus any of these others? Is there any advantage to using one versus the other? Which is better? Those types of questions. The answer to those questions, the basic answer to those questions is it's really a matter of personal taste. And I'll reduce it down here for you. All of the map, right? All of these algorithms that have sort of the map suffix on them, they are said to preserve the global structure of the data. And all that means is that they will juxtapose cells that are more highly similar to one another in two-dimensional space. So they'll put them closer together, right? So for example, here we can look at the CD56, which is this kind of bright green population, the NK cells, right? It pits them fairly close to our CD8 population over here. Likewise, it also kind of pits them fairly close to the monocytic cells and the CD19s, but they're not very close or not very highly related to these CD4 T cells, right, which are essentially across the ocean here. In all of these images, that is the case. So the more distance that exists between these islands or these blobs, the general idea is that those cells are going to be less related to one another, and that they don't share a ton of markers in common. Now, in contrast, t-SNE doesn't do that. t-SNE doesn't care about the white space insofar as it doesn't try to put groups of cells that are very close to one another necessarily in t-SNE space. So for example, again, here looking at the same data set, your CD4s are in red here, but you can clearly see that there are populations of this CD56NK cell subset that are very close to the red blobs that you see kind of going down through the middle. Okay, so just by glancing at this, you would think if t-SNE was preserving global structure that, oh, NK cells and CD4 T cells have a lot in common, when in reality they don't. Okay, so that's the sort of difference in terms of the projection here and what they do. Now, I recommend most people, when they use UMAP or any of these others that have the map suffix, you'll notice that they have these sort of peninsular projections coming off some of these blobs. And these peninsular projections are kind of nice in that they give you this idea that there's a group of cells that belongs to this much larger body. They're highly related to this much larger body, but these guys are breaking off because they have a marker or two that are changing. And so a lot of times you can infer trajectory or development, let's say, of a particular cell type, is moving along some sort of trajectory, and it's easier to see in these map type projections. Now, amongst the maps, which one you choose, I don't know, it's kind of up to you. To me, they all perform a very similar function. Maybe TriMap or PackMap has a little bit more detail in terms of the the complexity of the population versus UMAP here. And what I mean by that is, if we look at UMAP, we can kind of see one big blob here and one little blob that's off to the right that kind of describes our CD8 T cell population. But if you look in the TriMap and the PackMap, you can kind of see that, it looks like there's actually 4 projections. So there may be, I don't know, 4 slightly different populations of CD8 T cells present rather than just 2. Now, TISNE, as I mentioned, doesn't do a really great job of juxtaposing the very similar cells to one another. But what it does, or what I think is great about it, is that the granular differences that exist between cell types are displayed really, I think, really quite clearly. And the cool thing about this is if we look at UMAP, for example, and again, we look at the CD56 population, we can see that there's one major, CD56 population present with maybe 1 or 2 extra peninsular populations here. So you may say, okay, there's maybe 2 or 3 NK cell subsets in this data set. But when we look at TISNE, we can count 1, 2, 3, 4, 5, 6, 7 different populations of NK cells. So for whatever reason, TISNE has broken this population up quite significantly and spread it across the map. So in theory, these populations of NK cells are different than one another based on at least one marker. So even though they don't juxtapose all the NK cells sort of together in the same space, it does break them up across the map so that you get the idea that you may have multiple phenotypes present, where that is kind of lost in the UMAP or even in the PackMap or TriMap displays. So generally speaking, my favorite algorithm to use is TISNE for this purpose, because the granular differences appear a lot easier to see than in the other projections. But again, it kind of depends on the analysis that you're going to do. If you're going to do some, trajectory analysis, development analysis, maybe the PackMaps, TriMap or UMAP is better. I like to use TISNE when we're comparing conditional differences, right? This is treated versus non-treated, healthy versus disease, et cetera. I can see all the phenotypes compared to one another in this space, and it'll break it up for me so that I have the inference that there is something significant that is different between the two. Right now, when it comes to clustering algorithms, there are several clustering algorithms that we have available. These are just 3 of maybe 5 that we have available. They're my favorites for different reasons. FlowSum actually comes with a visual display. I don't really use this very much, but it is kind of a nice addition to just having populations spit out in the workspace for you. Phenograph is probably my favorite algorithm because it has a higher fidelity in my hands anyway when I'm applying the algorithm and sort of comparing it to manual gates, right? I have my manual gates that describe different populations of cells. I use Phenograph, and then I want to see, does Phenograph recapitulate what I find with my manual gates, yes or no? And so I find that Phenograph does a really great job. XShift is also a good algorithm. It's very fast. Takes probably the fastest algorithm that we have, so it'll crunch the data very quickly. But in any case, all of these clustering tools can be used with UMAP, as I'm showing here. The populations are just being overlaid with the UMAP plot that you saw on the previous screen, and it's dividing up the populations based on what the algorithm sees. So we'll see how this works out in the end. Now, what I will show you is how to phenotype all the different, populations that the clustering algorithms spit out. We use another tool called the Cluster Explorer. I'll jump into this in quite a bit of detail later on. This is an extremely powerful tool, but it basically presents you with a series of plots, one of which is a line trace plot that has all of the markers on the x-axis and the relative level of expression on the y. And you can gate on this to filter out cells that you're looking for. So you can identify various populations of cells. Those can be back mapped onto your UMAP or t-SNE, whatever you want. And of course, you will have a standard heat map for the MFIs of those populations. In particular, I like the Cluster Explorer for doing this particular function that I have outlined in orange here, and that is the ability to quickly compare two different experimental conditions or more to each other and have those conditions sort of heat mapped, so you can focus on the clusters of cells that are different between your conditions. And so we'll get to that. We'll get to that in a moment. For the last part here for tracking, we're working on an algorithm called TrackSum. I'm not sure if you guys have seen this paper, but it's kind of a cool paper that shows how the populations of cells are changing over time or disease condition. And it allows you to visualize, whether or not one population of cells is becoming many, right? The many different cytokines being expressed, as the disease becomes more severe, or are those populations sort of remaining fairly steady across the course of disease? And as in the case of the neutrophilic population here, and maybe the plasmacytoid dendritic cell population, but as you can see, the monocytes, which are kind of outlined in green here, they split and become multiple different populations of cells throughout the duration of the experiment. And so there's a lot of neat little ways to display the data within TrackSum. We're trying to get this into a plugin, as I mentioned. So hopefully that'll be available fairly soon for you guys to test out. Okay, so let's actually look at a practical example here and work our way through it based on what we have learned in the workflow portion of the webinar. Okay, so here I have 4 different files from 2 different patients, and essentially this is a fairly simple example, but it's I don't know, 13 color data set, and it's complex enough that we can use the dimension reduction and clustering algorithms with, and it also has we have a treatment associated with it, so I can kind of show you a lot of different manipulations within the program to help you analyze the data and hopefully get you started on analyzing your own data using these tools. So as you can see, we have 2 samples that have not been stimulated, and then 2 samples that have been stimulated here with the toll-like receptor agonist. And what I would like to do is annotate the data files here in a particular way so that when I concatenate them together for t-SNE or for clustering, those annotations will appear as gateable parameters in the concatenated file. Okay, so that we can make cross comparisons between stimulation and not, or patient 1 versus patient 2. So when you are at home or at work, and you're looking at your data and you want to annotate it for analysis in high D, what I would recommend is write out all the different experimental variables that you have present in the data set, and then add those as keywords to your data. So the first thing we're going to do here is we're going to go ahead and annotate the data by adding a keyword that describes something about the experimental conditions that we have, right? So we have a stim versus no stim. So I'm going to call this particular keyword header just the stim condition. And then the values that I'm going to assign here are going to correlate with whether or not these samples have been stimmed or not. So what I recommend that you do here is that you use non-zero integers for the designations. Okay, so non-stimulated, we're just going to assign the value 1. We're stimulated with the TLR agonist, we're going to assign it the value 2. If I had other files in here that were maybe stimulated with LPS or IL-1 or TNF or something like that, I would just continue to add numbers to the list. So if it was LPS, then I would assign 3. If it was TNF, I'd assign 4, blah, blah, blah. Okay, so annotate the data using numbers here to describe the different conditions. Okay, so we have 1, 1, 2, 2, right? The 1 corresponding to the non-stim, the 2 corresponding to the TLR stim. All right, now we have another condition here that corresponds to the patient number or the patient ID, right? So you can see here is PID1, PID2, PID1, PID2, right? So we have 4 files coming from 2 different patients. So we're going to add another keyword that describes the patient ID or the patient number. Okay, so we add that, and then again, we're going to use integers to describe this. So we're going to say the patient number 1, we're just going to assign the value 1. Okay, and then patient number 2, we're going to assign the value 2. The sample comes from patient 1, we assign it the value 1. If it comes from patient 2, we assign it patient 2. Okay, you can add a plethora of these types of keywords to the data set so that you can make comparisons downstream between the different stim conditions, the different patients, maybe a stim condition from patient 1 versus stim condition from patient 2, etc. Okay, so create these keywords to annotate the the different experimental variables that exist. Now, these data have already been compensated and they've actually been cleaned up. We have a lot of cleanup gates that have been added to the data set. So we don't have to create those gates. We have a lot of downstream manual gates as well. Okay, so the annotation step is done. Now what we want to do is we want to consider something that I didn't discuss in the workflow there, and that is downsampling. So maybe I need to add that as a bubble. But downsampling is a step where we reduce the numbers of events down to some arbitrary number of cells. And the reason that we do this, at least the primary reason, is that TISNE and UMAP and all these kinds of tools, they tend to be a little bit slow. The clustering algorithms are fast, but the dimension reduction tools are usually fairly slow. So what I would recommend that you do here is you take a look at the population that you want to concatenate. So again, we clean up the data, and what we want to feed to the algorithm is something downstream of the cleanup gates. Right, I've got a dump channel here, and then I have essentially the antigen presenting cell compartment and all of its downstream populations. So it's up to you where you want to begin the analysis. I like to start, kind of high up on the hierarchy, as high as I can go after the data have been cleaned up so that I can see all the different cells projected onto my TISNE space. Okay, but if you guys are, I don't know, monocytic specialists, perhaps you just want to deal with CD14 positive cells, or if you go even further, maybe you want to just deal with the patrolling monocytes. But whatever the case may be, pick your population that you want to begin the analysis at, and then look at this population across the other samples that you have, right? So we're going to expand this and look at how many cells we have for the DR positive, right? So we've got 135,000 in sample number 2. In sample number 1, we have 130,000. Then when we get to the TLR stim and we look at the DR positive population, suddenly we have a steep drop off, right? We only have 28,000 cells here in the 3rd sample. And then when we get to the 4th sample, also TLR stim, if we look here, we can see we only have 21,000 cells. So you have about a 6 to 7x difference in the quantity of sample number 2 versus sample number 1, or sorry, sample number 4 versus sample number 1. And if you don't normalize the numbers of events, it makes direct comparison between those samples difficult, right? So if you have 7x less cells here, and then you go in, you just concatenate everything together without downsampling, you're going to have 7 times the number of cells represented for patient number 1. And so when it comes to frequency differences between populations, you will always have to remember that, in the original unconcatenated file, there's a 7x difference between these samples, and it's not due to the treatment, it's just due to the fact that I had fewer cells. So in order to sort of avoid that headache of having to come back and think about how many cells you had, I would normally pick the sample that has the fewest number of cells and then just downsample everything to that particular level. Now, in today's webinar, because these tools are so slow, I'm going to reduce the numbers of events to be even less than that. So what we can do here is we can select the DR positive population. This is the level we want to begin at. And we can go to the workspace tab and we can use the plugins drop-down menu here to find our downsampler. So this will just, again, arbitrarily reduce the numbers of events to some number that we choose. The default here is going to be 3,000. So we're going to change this. I'm going to reduce everything down to 10,000 cells. And we can just call this, I don't know, DR down or something like that. Click OK, and it's going to go ahead and downsample that population. It'll end up at one level indented underneath that DR population, and you'll see here that we have 10,000 cells. Okay, now I want to do that for all the remaining populations that I have present. So what I'm going to do is I'm going to copy this downsample down arrow, this little plugin node as we refer to it, and we're going to just drag and drop it onto the DR positive population. Okay, and when we do that, now we should have downsampled DR populations across all of the samples. Okay, so there we go, our 10,000 cells. Now what you can do is you can select them all, right? Because we want to concatenate them. Remember, we have to merge these guys together before we can do any of the fancy stuff. So we're going to right click here and we're going to say, select equivalent nodes. Okay, and then what that's going to do is it's going to highlight all of the DR down populations that you have, okay, in the workspace. And then what we're going to do is we're going to concatenate them together. So I right click on one of them. It really doesn't matter which one, once they're all highlighted, and then I say export concatenate. And this is going to bring you up to our concatenation window. Now, the first thing you have to do is make sure this is on concatenate and not on export. It defaults to export. So you want to kick it over to concatenate, make sure that blue highlight is either within the box or around the concatenate. Make sure you know where the file is going. Do not concatenate onto a server location. I would concatenate and dump, directly onto your desktop, for example, which is what I'm going to do here. Okay, you can say include all events because we've already downsampled. This little box allows you to downsample. If you don't use the downsample plugin, you can do it here if you want. Okay, now for parameters, you probably want to use this option where it says all compensated parameters because you want to preserve the compensation matrix that exists on all the different files that are present. So if you have files that come from different days, they have different compensation matrices, and therefore you want to preserve the compensation that is on each of those files when they get concatenated. And the way to do that is to choose this option, all compensated parameters. Okay, now it just so happens that today's experiment that I'm going to show you here, all of these files were compensated on the same day. It's the exact same compensation matrix. So I'm going to just choose all uncomped. And then what's going to happen is once I, concatenate the file, it'll fall back into this workspace and it will pick up the compensation matrix that's associated with these files. But again, my choice here is the exception to the rule. You guys are going to want to use this choice here. Okay, so I am doing something sort of strange in that I'm choosing that option. All right, now for the concatenated file, you could call this, give it a name. I usually give it something that kind of denotes where or what I did with the file. And then super important is to make sure that your additional parameters, the keywords that you added in the workspace here, right? These guys on this side over here are included. So you click the choose box, and then we scroll down to the bottom to find the keywords that we added. So we added something called a stim condition, and then we also added something called the patient number. Okay, so then we go ahead and choose those, click OK. And it should mention here that you're going to have 2 keywords present. All right, so then from there, what we can do is now we're ready to concatenate or merge the files together. So we click concatenate, and then you can tick the box for dumping it into the existing workspace. You can put it into a new workspace. You can do both. You can do neither. It's up to you. Okay, I'm going to put it into the existing because I have a group here that catches concatenated files. So you can see here that the concatenated file is present, and it picks up the compensation matrix that was associated with those files. Okay, and hence all of the downstream gates. Now I'm going to get rid of the downsample node here just so that we don't have any recalculation when we run any of the other tools. So it's good after you downsample, good idea to just get rid of that node so that you don't have any funny business when you're trying to do other things. Okay, now what I want you guys to do at this step essentially is you're going to want to gate your stim condition and patient number parameters. I do that first so that I can see all the comparisons that I can make in the data. So open up the graph window, you change the x-axis over here to one of those conditions. You see how those keywords became conditions, right? Or those keywords became parameters, right? There's my stim condition, there's my patient number. So maybe I want to start with my stim condition. And you can see I have 2 populations, basically 1 and 2. And this corresponds to, right, patient or the non-stim condition and the stim. So normally what I do here is I just change this over to like a histogram. I'll make this linear, and then I'll create a gate, right? And just draw, click and drag across. The first population is the non-stim, right? And the second one is the stim. Okay, and when you draw these gates, it's a wise idea to just go back to the workspace and check that the gates that you've created include as many cells as they're supposed to have. So remember, we downsampled to 10,000 cells a piece. The concatenated file had 4 files, so we have 40,000 total. When we split this up into non-stim and stim, there should be 20,000 a piece, and that's what we get. Okay, same thing should happen now when we gate on our patient numbers, right? So we change the x-axis here over to patient number. We'll go ahead and linearize the axis and do the same thing, right? We say patient, or just PT1, that'll probably be sufficient, and then PT2 for the other. Okay, check that our numbers of cells are good, 20,000 a piece for each patient, that's good. Okay, and now if you guys want to essentially recapitulate what you had in the original data set, you can take your stim gates and you can just drag them underneath, right? You can drag them underneath each patient. So now what you have, you've reconstructed all the different, experimental variable comparisons that you could have present, right? You can have patient 1's non-stim condition, right? Or patient number 1's stim. Right, patient number 2's non-stim and patient number 2's stim condition. Okay, so you just basically broken the concatenated file back out into its individual parts, but instead of being individual files, they are now populations. Okay, now that you've done that step, you're ready to do some fancy stuff. So I'm going to use t-SNE, but again, you could use UMAP or some of the other maps, right? So for example, you have UMAP here, there's TriMap, I have another one called PacMap. UMAP is a little bit slow in comparison to t-SNE, so that's why I choose t-SNE here, in addition to showing me all the granular differences between populations. So I will show you the t-SNE plot here. Maybe if we have time, we can do a UMAP as well. All right, when you come into the t-SNE prompt, just deselect the colors that you've already used to clean up the data. So for example, we have a dump channel that includes essentially all the lymphocytes. So you probably don't need that since we're dealing with just the antigen presenting cell compartment. So you don't really need the dump channel fluorochrome present. And you don't need the viability dye present either, because we've already cleaned up the data. So viability dye is not going to help us in any way to resolve these populations, okay? But you can keep all of the other lineage markers and cytokines that you have in the data set. Okay, and then the learning configuration here, I recommend that you use the opt-SNE. This algorithm is an improvement made by Anna Belkina at the Broad Institute. She essentially made the t-SNE algorithm move faster by adding in a step where the algorithm essentially monitors this so-called elbow plot to see the variance versus the number of populations that are present. And when it reaches an optimal position on that elbow, it basically stops the number of iterations from moving forward and thus makes the time truncated. So it's good. It gives you just as much resolution as you would if you didn't have the opt-SNE option. And I think it's a fantastic improvement on the speed of the algorithm. Okay, the number of iterations here, if you have less than, say, 1 million cells, probably don't need to play with this number. If you have more than 1 million cells, just bump it up to the max. Again, auto or opt-SNE will take over and will truncate the number of iterations that the algorithm has to go over. The learning rate here, generally, the paper states that this should be between 8 and 14% of your total number of cells that you're trying to crunch it over. So in our case, we have 40,000 cells. Rather than trying to calculate what 8 or 14% is, 10% is between 8 and 14. So that's easy. Just tell it to do it with 4,000. The perplexity is a knob that controls the white space essentially that exists between the islands. If you need more white space, you may want to, take the value up. However, I will say that perplexity is kind of a double-edged sword. Sometimes you can add too much value here. The islands will just essentially slam back towards one another. The default value here, I think, is pretty good. You can play around with it, though, moving it up or down to get higher resolution in your t-SNE plot. And then the KNN algorithm, there are two that you have available to you to choose from here. The exact vantage point tree is a slower algorithm. The Annoy is faster. This is actually what Spotify uses to predict the next song in your playlist, especially if you hadn't heard it before. So I think Spotify does a pretty decent job of identifying songs that I like anyway in my playlist. So I'll go ahead and use that. Then for the gradient algorithm, the original published algorithm is Barnes-Hutt, but there was a modification made in 2010, maybe 2011, called the fast Fourier interpolation, FITSNE, and it moves a lot faster. So if you want ultra fast t-SNE, these are the settings that you would apply. And then go ahead and click the run button here when you are ready. And you'll see in the upper, well, in my case here, the upper left, you can kind of see this little blue bar extending across the screen, calculating t-SNE. Okay, now it's going to take a few seconds here for us, maybe 30 seconds to complete. So I'm going to check the chat box real quick. And I don't see anything there, so we're okay. But if you have any questions, by all means, go ahead and ask. And as I said, if we have time, we can probably do a UMAP, and maybe I'll run the UMAP after we run the TISNI, and then we can do some fancy stuff in the layout editor while the UMAP is calculating. And then you guys can make the decision for yourselves which one you prefer. Okay, so there's my TISNI plot. Don't have a ton of different islands, right? They're not super well separated, but sufficient for what we're going to do today. We've got a big blob over here, another one over here, another one here, and a few other islands present as well, you know, somewhere in the middle, etc. Now, when you get this initial TISNI plot or UMAP plot, it's not abundantly clear, you know, what these islands are. So there's a couple of different ways to sort of pursue or define what the islands are. You could do it by, you know, opening up the layout editor, for example, and just dragging in your, you know, dragging in your TISNI plot. So let me get this workspace on this side, and then we can just drag in the TISNI plot fairly easily here. Okay, so we can take this, dump it here, and then there's a quick tool. If you right click on the TISNI plot or the UMAP, say make multigraph overlays, there's this option for multigraph color mapping. Gives you a little prompt where you can choose all your compensated parameters, again minus probably the viability die because it's meaningless, and the other one, dump channel. And then it'll take a second or two, but what it'll generate here for you is a nice little panel that has your TISNI heat mapped by marker, right? So you see this a lot when you go to trade shows or, you know, conferences, and you'll see that you know, people have a poster that has, you know, these designations. But in any case, this will help you to identify the population of cells. So for example, I can see where my really highly pro-inflammatory IL-1 expressing cells are located on the TISNI plot. You know, if I look at CD14, for example, I can understand from this that, you know, this lobe on the bottom left has some my monocytic cells, right? Maybe this island over here and this island as well. The other way to do it is to just simply take your TISNI plot, and what you can do is you can overlay your known populations on top of it. So for example, I can overlay my, you know, CD14s, I can overlay my, you know, dendritic cells, and I can overlay, for example, my PDCs here, right? My plasmacytoid dendritic cells. You can overlay all of these guys and then just change the axes here so that we're looking at, right, TISNI 1 and TISNI 2 as a, I like to look at this as a contour, but maybe as a dot plot initially, it'd probably be the best. So we'll do TISNI 1 and TISNI 2, click OK. Right now you can kind of see, oh, okay, that's where my, you know, where my different populations that I've predefined, where they exist on the TISNI plot, right? So my CD14s are in blue, right? My dendritic cells are in orange, and then my PDCs are in green over here. So that's all cool, but what we really want to do with this is we want to make comparisons between the samples. So let me get the UMAP started, and then we can start making comparisons. So UMAP has the same sort of interface, right? It's got this parameter selector where you're going to have to choose all your compensated parameters. And again, we're going to feed it everything minus the dump channel, right, and the viability die. There's some boxes here that you can tick. Nearest neighbors is going to help to separate, again, the number of islands. The lower this number is, the more islands you're going to get. The higher the number is, you're going to get more conglomerates. It's kind of up to you how you want to do it. And you can also change the distance metric that it uses. Euclidean is what the paper published, but there are other ways to measure distance between populations in multidimensional space or hyperspace. And I'm not mathematically equipped to describe all the other methodologies. But in any case, go ahead and click OK here after you choose that, and UMAP will get underway. Okay, now, as I said, we want to make comparisons between our conditions. So, for example, if I want to just see what the difference between the non-stim condition is and the stim in both of these patients, I simply overlay the two. And then again, I'm going to go ahead and double click on the plot, change this to maybe a contour. And then we're going to do t-SNE1 versus t-SNE2, right? Click OK. Another thing we might add to this is showing the outliers so that you can see if there's any cells that are in different positions, etc. Now I'm going to zoom in here so that you guys can see this a little bit better. But the bottom line, and then this is what I think makes this tool so cool, is that you can see the difference immediately between the non-stim condition, which is in red, and the stim condition, which is in blue. So the blue islands, right, the blue areas, are groups of cells that are phenotypically distinct from the red or the non-stim, right? So these are new phenotypes of cells because they have no overlap with the red. Okay? In contrast, there is an island up here, this big blob kind of at the top. You can see that there are blue contour lines and red contour lines, and this is telling you that there is overlap, right, there between the two. Now our UMAP has finished, so we have that as well to use if we want. But in the contour plots here, you can see that there is, the fact that you have red contour lines and blue contour lines in the same space is just telling you that these cells are the same phenotypes between non-stim and stim, but the frequency between those two conditions is different. And that's kind of implied, again, by the number of contour lines that are present. So whatever these cells are, they're in much larger abundance in the stimulated condition versus the non-stim. Okay, and obviously there are cells here in the non-stimulated condition that are not present when you stimulate the cells, right? Basically, once you stimulate the cells, they get angry, they start producing cytokine, and they sort of become these different functional states of whatever these cells happen to be in the beginning. Okay, so now the question becomes, okay, well, I can see phenotypic differences between these two conditions. How can I phenotype them, right? How can I ID what the phenotypes are between the different conditions? So one way to do that is just to create gates on the populations that you find interesting. So for example, if I like this little guy right here, I can simply create a gate on this and then overlay it here and use another tool to be able to visualize the phenotype, right? So the way to do this, since I'm looking at a blue population, right, that corresponds to the stim condition, I can go back to our workspace, go to the stim condition here, right? And I can basically create a gate on this. So we have to put this back to t-SNE. All right, t-SNE1, t-SNE2. And again, I like to make it look like the contour that we have, show the outliers, something like that, right? And then we can just simply create a gate on the area of interest. Now, normally I juxtapose the graph in the layout editor so that I know what I'm targeting. All right, so over here, let's say I'm interested in this guy. Okay, so that corresponds to this little population of cells there. So I take my little gate and I create a polygon around it, right? And I can just call this, I don't know, pop with a question mark, maybe query, whatever the case may be. And then you take that population gate and you drop it on top of your overlay that you have set up. Now, I recommend that you change the color here to something, very strong and contrasting with the white background. And then from here, you right click and say make multigraph overlays. And this time you say histograms. Okay, and then the histogram panel will appear and you'll have to stretch this guy out quite a few times in order to get it to, like, be nice and easily visible. So I like it kind of tall but slightly narrow. But it's really a matter of personal taste. Okay, so we get it big enough here so you guys can see. And then I like to trim this list because you can see that there are uncompensated parameters here and they're not relevant to our analysis. So I right click on the panel, I go to the very bottom and I just say select multiple. And this allows me to tell the panel like what to actually show me. Okay, so I want everything again except the viability dye and the dump channel. Click OK, and that will trim the list for me. Okay, so now what I can do is I can look at the line trace in these histograms to try and figure out what the phenotype of these populations happen to be. So in this case, this population expresses to some extent IL-6. It's obviously very high with respect to HLA-DR, right? We have a decent bit of CD1C. So this is a dendritic cell marker. So it's probably a pro-inflammatory dendritic cell that expresses 83. So it's relatively mature. It expresses a lot of IL-1. Okay, so it's also, again, pro-inflammatory that way, but it's negative for most of the other markers. Well, it's got some IL-12 that's present as well. A little bit, not a ton, I guess. Okay, but we come back to the gate here, right, our little pop gate. And if you push the up arrow, you get back to that graph that you were working on. And what you can do now is you can explore all the other populations of cells that you have present here, either by creating more gates and then overlaying them, just as I have done here, or you can simply roll this guy over and it will update your little histogram that you have here. So here, these cells are very clearly IL-6 positive, still CD1C dendritic cells, got a lot of DR. These guys express TNF, unlike the brothers that are right next door that are TNF negative. It's got a lot of 83, so they're still mature. They got IL-1, right? And they have a lot of IL-12 present as well, okay? But negative for essentially everything else. So these guys are pretty super inflammatory dendritic cells. I don't know, we come over here. I think these were the PDCs. So if we actually look at these guys' profile, they have 83 present. Yeah, there they are. They're 123 positive as well. Okay, so this is sort of a qualitative way to look at the, at the populations that you have there. Now, if you're interested, you can always add statistics. So if you were rolling that population around and you want to derive statistics, I usually tell people just, make use of the shortcut over here. And say all compensated, and you get a nice little panel with all the MFIs. And as you roll this around, these MFIs update, so you can kind of track it, quantitatively if you want to do that. Okay, so we can take a quick look at our UMAP. Come over here, drop this in. Same thing. We would change the axes here to UMAP 1 and 2. All right, so UMAP, there we go. This is our UMAP plot. You can juxtapose this with your, t-SNE plot if you want. So you can duplicate this. Duplicate, there it is. And then just, put, I don't know, t-SNE 1, t-SNE 2. The UMAP in this case looks a little bit better than the t-SNE did. Okay, and then of course you can do, I don't know, if you want to look at the population overlays, where did we do that? Right here, right? Duplicate this guy. Thank goodness for the undo redo, right? Duplicate and show the UMAP here. And you can kind of see, again, how does this compare? Does it look better or worse in UMAP versus t-SNE? Right? UMAP did a better job, I think, this time of segregating these monocytic populations out versus, versus TISNE. It's not well resolved. PDCs are okay, and your dendritic cell population seems pretty clear in either instance. Okay, but in any case, again, they're just, differing dimension reduction tools, kind of a preference, personal preference, how you want to pursue it. Okay, and you can do the same thing that we did here in the TISNE plot. We can do the same thing with UMAP, right? So once that's done, you can essentially, look at the same thing on the UMAP parameters. Okay, and if it helps you to ID those cells better, then use UMAP, or any of the other dimension reduction techniques for that matter. Okay. All right, so that's kind of a simple use of TISNE or UMAP, the dimension reduction tools. But as I mentioned, you probably want to run this in conjunction with clustering algorithms. So we're going to go back to the plugins drop-down menu, and I'm going to go ahead and run Phenograph. I like Phenograph again because I think it does a really great job of divvying up the populations that are, it's more consistent, I think, with manual gates than the other algorithms are. But I mean, it's a very narrow margin. So anyway, the K value here, again, if you have a lower K value, you're going to have more clusters. If you have a higher K value, you're going to have fewer clusters. So I normally start out this with around 20, and then I go from there. If it over clusters or the model is overfit, I can always merge the clusters after the fact, after I figure out what they are. But it's better to over cluster or overfit the model than it is to underfit, especially if you're looking for rare populations of cells. You want to break up the data in as many pieces as possible so that you make room for that rare population of cells. If you underfit the model, you may never see those rare cells unless you know what you're looking for. Okay, so Phenograph finishes, and as I mentioned, we can kind of try to identify what these phenotypes are by, dropping them into the layout editor. You could do the same thing like we did before, where you take the, different populations, you overlay them in the, UMAP or TISNE space. Let's put it in UMAP this time. And go here with UMAP as well. Right, you can see how Phenograph kind of cut up the different populations of cells. Now, I don't want to have to go one by one, especially if I have to use the histogram tool to try and figure out what these populations are, right? I might have something set up like this, put this on this side, and then I go over here and I say, okay, well, now do, histograms. This is going to be a really hairy method to try and figure out what each of the phenotypes of those clusters, are, right? All the different line traces, it gets really complicated very, very quickly. So we rely on another tool called the Cluster Explorer to figure out what the phenotypes are that, Phenograph is spitting out for us here. And it gives us the advantage of being able to show which clusters are appearing in one condition versus the other, which is ultimately what we want to know anyway. Okay, so once you have your clustering algorithm finished, you can choose, whichever clustering algorithm you want. Go to your concatenated file and then select the plugins drop-down menu. And now we want to go to Cluster Explorer. Okay, same thing. You have a parameter selector here. Deselect the viability, pardon me, and the dump channel. And then this is going to open up a series of windows for you. So sometimes it may take a little while to open up, but have patience. It will show up, and there's your heat map, right? The line trace plot. And then you have your bar chart plot over here that has all the different clusters and the relative numbers of cells that it has in each of those clusters. You have the control panel that's set over here, and then your You have a singular 2D plot if you have one dimension reduction done. If you did two different dimension reduction techniques like we did in ours, we have two. Okay, and then the cool thing is in these plots, you can gate on the parameters that you want to filter out. So for example, if I want to identify my dendritic cell populations, those that are CD1C positive, I'm just going to go ahead and create a gate, maybe on the ones that are relatively high for CD1C expression. It'll filter out all the other clusters, and then you can see here in the heat map, they're sort of each cluster is back mapped. They're back on this plot, and they are back mapped to your t-SNE and your UMAP space as well. Okay, you can further filter your markers here if you want by creating a second gate. So for example, maybe you want to go after that mid-expressor, right? So here's a dendritic cell that is mid-expressing for IL-1. Right, we've identified that as cluster number 11. Okay, and again, it back maps that onto your t-SNE or your UMAP plot. If you don't want to look at UMAP or t-SNE, let's say t-SNE was not particularly useful for us, and we want to, we can look at this population across any other set of parameters, right? So you could say, okay, well, show me this guy. You say that it's CD1C positive, so let's see what it looks like in terms of CD1C as well as, let's say, IL-1, right? You can always change the plots here, and you can add more. So you could just say, okay, well, that's cool. I want to see that one, and then I want to see another one. Yeah, let's say DR versus IL-6, or maybe we want to see, I don't know, 14 versus 6, right? And you can see that this population is negative for both of those markers. Okay, but IL-12, I think, is popping up over there. So let's see what it means by mid-expressing of IL-12. Okay, maybe a little bit, mostly negative, but there are some cells here that on a periphery that are probably positive. So again, this could afford to be, we could overfit the model a little bit more to try and tease apart this population of cells over here away from the bulk. But in any case, you can see those cells presented to you. Okay. Now in the control panel here, you can choose which clustering algorithm you want, which sets of clusters you want to see. In our case, we only did one, which was phenograph. But if we had FlowSum also done, you would see a line trace here where we can intermix between phenograph and FlowSum. You can change the logic on the gates. So you can, essentially change this. Let's clear the gates and create I don't know, we'll just create another one here. We could say, I want to look at this and not, these guys, and it'll actually use Boolean logic to filter out. Okay, you can change the profile points here to plot medians instead of means. And you can use what are called channel values rather than scale values. Scale values are essentially what you see in the plots here, and channel values are the raw values that are sort of the raw data bins that are behind the data that you see in FlowJo. All right, so that's the control panel. Now for the ultimate, I think the ultimate tool here, besides this line trace plot, let's go ahead and clear the gates here. It's the heat map. As I mentioned, you can modify the heat map so that you can look at two different conditions and see how the populations skew one way or the other. So I would go to the options drop down here and change this where it says add nodes as columns. So you click on this, and then from here, you can go ahead and select your stimulation conditions, right? So you had non-stim versus stim. You could choose patient 1, patient 2. You could even do stim from patient 1 and stim from patient 2, whatever you want. But basically, you're adding your categorical variables here. And then what happens is you get a new heat map that is showing you the difference between those two conditions, right? So in the case of the new heat map, The yellow means that there are more cells essentially in this condition, the non-stim in this case, than there are in the stim, right? The yellow meaning very high, the blue meaning very low. So what this is telling us is that clusters number 2, 3, 4, 5, and 6 are more highly associated. These are populations that are coming from the non-stimulated condition versus the stim. And if you hover your mouse over long enough, it will actually tell you what the breakdown is. So you can see here, there was, I don't know what, 3,300, 3,200 events. 98% of the cells in cluster number 2 are coming from the non-stimulated condition. Okay, same thing for some of the other blocks or other populations here, right? Most of them coming from non-stim. Then when we get down further here, right, we see some yellow blocks in the stim condition. And of course, here we can see that, okay, cluster number 17, you've got a pretty heavy split again, right? 90, basically 100 to 0, right? Another one, 97, 98 versus 2, right? And 99, again, 100 versus 0. Okay, and then as we move to the colors that are a little bit starting to cool off, you can see that that percentage now becomes like 90-10, right? Here's 80-20, let's say, right? This one's pretty even, 50-50, right? Again, another non-stim cluster, so on and so forth. Okay, so but the point is, this particular setup here allows you to quickly identify how the populations are skewing one way or the other. So if you're only interested in the stim or the disease condition, you're going to focus on the clusters that are not, the non-stim. So you're going to look at everything from cluster, let's say number 7, all the way on down to 19, right? And you're going to eliminate all the other populations. Maybe you want to look at cluster 1 here because that's a 85-15 split. Okay, you can use the heat map if you want, or you can just simply select the cluster here. It'll back highlight onto, again, the plot or in your line trace. And as I said, cluster, I don't know, let's say 7 through 12 were the sort of mid expressors, minus cluster number 10. All right. So you can see how they get mapped here, and you can see the difference between them. Now, again, I mentioned that clustering algorithms are really good at picking out the mid-expressing cells, and you can kind of see that here. There's some mid-expressing CD1Cs. Oops, let's do an and, clear the gates. All right, there's some mid-expressing CD1Cs. There's also mid-expressing for TNF, right? 83, same story. You have mid-expressors for, right, 83. You obviously have mid-expressors here for CD14, IL-1, right? So these are populations of cells, again, that I'm not sure that I would easily gate them by eye because they have a slightly different distribution that I'm used to, especially if I'm just gating on-off for most of my fluorochromes, right, including the cytokines. So it's just a good way to kind of review the data and make sure that you're not missing anything at the very least. Okay, I'm going to close the Cluster Explorer, and I'm going to show you the last part of the sort of the analysis that I think is important here, and that is, let's say that cluster number 7 was really interesting to us. It was associated with the stimulation condition. We don't know exactly what it is, and we want to now create manual gates to identify this population. How do we create a series of manual gates for an algorithmically derived population? Okay, same thing with our t-SNE gate that we created earlier. If we found a population of cells that we're really interested in, how do we get back from the, from t-SNE, how do we translate this essentially into useful gates? So what you need to do is you select your population, you select your concatenated file, and you use a plugin called Hyperfinder. And what this thing does is it essentially asks you what series of parameters you have available to you to gate on. Maybe again, minus the viability die here. It asks you the number of parameters, or the parameters that you want, how many gates you can have to create that population. Now, the more gates you have, the more specific and the more granular this tool can be. The number of vertices, if you have 16 vertices, that's pretty good. I wouldn't increase it any further unless you absolutely need more precision, but this is usually pretty decent. I can't remember the last time I created a 16-point polygon gate. And then the F measure beta here, I apologize because I always forget which direction this goes, but increasing this value skews the algorithm to higher purity, and decreasing it skews it to higher yield, or vice versa. Leaving it at 1 means that you treat the purity and the yield equally. Okay, but if you want to go more purity or more yield, you go up or down. And the documentation tells us, what that is, but I always forget the order. And then you just pick the population that you want to have described by the Hyperfinder plugin. So you can choose multiple or one, however you want to pursue it. I'm just going to choose phenograph cluster number 7 and say, show me how to get to this population using the gates that I have above. So it'll take a second or two, pardon me, and it will it'll create a series of gates here that it'll use to find that population 7 that you have in the workspace that's coming from Phenograph. Okay, so now I'm beach balling, unfortunately, but hopefully something will show up here in a minute or two. Okay, if not, I'm going to check back on our workspace and just make sure that I don't have any other plugins sort of roaming in the background, which I don't. Okay, so there's the image. And so what the algorithm does is it goes through several iterations, gating on your population of cells using the gates that you defined, or the colors, the fluorochromes that you give it to define this population, and then it'll give you this series of gates to use once it finishes to identify that population. Okay, so we're basically just translating what we had in the mathematical space over to manual gates. So again, here's my 8 gates that I told the Hyperfinder to find. Your population is going to be defined by hi-fi, blah, 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 and you'll see that it's phenograph number 7. And the easy way to sort of identify or look at those manual gates is just take in the terminal population into the layout editor, and then right click here and say, show me the ancestry. And if you want, you probably want to look at this, I don't know, maybe not as a histogram, but as some other set of parameters. But here, having trouble with my zoom, but in any case, right, you can see all the populations, all the gates. These are the manual gates that the Hyperfinder found for you to use, right, to identify that terminal population, that phenograph number 7 population that you were looking for in the end. Okay, and then if you want to sort on this, the cool thing is that you can highlight these, you can right click, and you can say export to BDFax Diva. So if you happen to have a Diva experiment already set up, you just pick that experiment, you pick the gates that you want to add, probably just want to add these guys and not all the others. But you pick the gates that you want to add and then say export to Fax Diva, and it will go ahead and modify the Diva experiment to include those gates. So if you wanted to sort on them, you could essentially get the rough set of sort gates for that specialized population of cells. Okay, so with that, I will take any questions that you may have. This pretty much ends today's advanced session. It's a little bit shorter than the other one. But nonetheless, I hope I've given you guys an idea of how to use these tools, what you can use them for, and I encourage you, to test them out. And if you need to get these plugins, like for example, if you want to get UMAP or Phenograph and things of that nature, you would go to the plugins drop-down menu here and click the very top option that says FlowJo Exchange. And then what this does is it takes you to our website here, and you can see all of the different plugins that we have. I'm going to kind of turn off the SeatGeek plugin here for the time being. But you have all the different plugins that are available. There's XShift. FlowMeans is another clustering algorithm. I didn't show that today. There's your hyperfinder. There's a cytonorm that I talked about initially. EmbedSum is really nice for very fast dimension reduction if you guys want to try that out. There's our TriMap. And then, of course, you have some other tools here to give you different renditions if you want a minimum spanning tree, maybe different ways to sort of graphically represent your populations. Euclid is a great tool for evaluating, how well the model is overfit versus underfit. Fate is a new dimension reduction technique. PackMap is down there at the bottom. Yeah, so they're all here, and all you have to do is download and follow the instructions to install the plugin. Okay, and if you want at this stage, you guys can also unmute your mics and just ask a question verbally if you don't want to type it into the chat box. And I think with that, what I'll do is I'll just stop the recording.